Interphase before meiosis matches interphase before mitosis, consisting of G1, S, and G2, and prepares the cell for division. During S phase, the uncondensed chromosomes / DNA are replicated and begin to condense by G2. The nuclear membrane encloses the chromosome-containing nucleus. The centrosomes are put in place by the end of interphase. By the end of meiosis, 4 genetically different haploid cells / gametes are produced, while mitosis 2 genetically identical diploid / SMAC cells are the end products. The partly uncondensed DNA condenses into the pairs of homologous chromosomes, with sister chromatids attached at their centromeres. 2 distinctions between meiosis and mitosis occur during this time: synapsis and crossing over. Each pair of homologous chromosomes in a diploid cell exchange genetic info, creating genetic variation. If every gene on these chromosomes were homozygous, then there would be less genetic variation, since crossing over wouldn't alter the alleles in the chromosomes. In the telophase cells wouldn't have distinctly different alleles. In most cases, they do. During this time, the nuclear membrane disintegrates, centrioles move to opposite ends of the cell, and spindle fibers form. In metaphase 1, the tetrads line up in double file at the metaphase plate, with each pair staying together. With the nuclear membrane disintegrated, the spindle fibers are able to elongate and attach the chromosomes at their kinetochores. The spindle fibers retract, pulling each homologous chromosome toward the opposite end of the cell of its pair. This is unlike mitosis, where sister chromatids separate. The centromeres still keep the sister chromatids together during anaphase 1. The cell itself elongates during this time, as it prepares for telophase 1. The diploid cell is now beginning the process of becoming 2 haploid cells. Cytokinesis overlaps with this phase. The cell begins to pinch in 2 and will become 2 daughter cells by end of cytokinesis. During this time, the nuclear membrane reforms and the spindle fibers break down. Unlike in telophase and mitosis, each sister chromatid remains attached at the centromere during telophase 1. This means that the number of chromosome sets in each newly forming cell has been reduced by half, and explains why the 2 cells are haploid. During cytokinesis, the cytoplasm divides, creating 2 genetically different haploid daughter cells. Each pair of homologous chromosomes is split up, with 1 homologous pair in each cell. Each cell has a nuclear membrane surrounding the chromosomes and centrioles. Meiosis 1 is now complete.
Here meiosis 2 begins and prophase 2 starts. There is no need for interphase because there is no need for DNA replication, as the number of chromosome sets is reduced from 2 to 1. Beginning of prophase 2, we see fragments of the nuclear envelope still present. What is also present is the spindle apparatus, which begins to form. As the spindle apparatus develops, chromosomes begin to move to the metaphase 2 plate. In metaphase 2, the chromosomes are lined up in a column, not in a row as seen in metaphase 1. The microtubules, which are extending from opposite sides of the cell, attach to the kinetochores of each sister chromatid. The bond between sister chromatids located at the centromere breaks, allowing the chromatids to be pulled apart to opposite sides of the cell by spindle fibers. The number of sister chromatids reduces by half in this division, and once separate, these chromatids become chromosomes. In telophase 2, the nuclei forms, the spindle fibers disintegrate, and the chromosomes decondense. Also in telophase 2, the cleavage furrow begins to form.
By the end of telophase 2, the nuclear membrane has begun to reform.
When cytokinesis kicks in, the cleavage furrow expands because the cytoplasm is dividing. By the end of cytokinesis, the overall outcome of meiosis is produced: 4 genetically different cells, gametes, with 2 haploid sets of unduplicated chromosomes.